If it could be demonstrated that any complex organ existed which could not possibly have been formed by numerous successive slight modifications, my theory would absolutely break down. Charles Darwin said about 165 years ago on The Origin of Species. And I think it's probably been a while, in fact, probably never a sermon has started with a quote from The Origin of Species. I can probably be sure to bet on that. But the reason I want to do that today is to challenge this, to take Darwin on his challenge. Because what he's saying here, essentially, he says that if somebody could demonstrate that a complex structure or organ could not have been formed by these three key elements, modifications that are numerous, that are successive, one after another, and that they are slight, each one being little in magnitude, then his theory would absolutely break down. And this is the challenge that I want to propose to you today for the next half hour or so. I want you to understand that we discuss here ideas and people's ideas, and while I will present people and their statements, it is not my intention to attack any of those folks. I am convinced that a lot of them are honest, sincere people. They present their views from where they are. And again, I'm not trying to demolish them. I'm questioning the ideas when I do so. The second thing I'm going to say here, because we want to get the main points, and this is a complex subject matter, I'm going to have to simplify some things. And for those of you who are more experts in these things, that may feel like an oversimplification, but it's for a purpose. So these things have a lot of bearing on who we are, what we believe, and where we ultimately end, spiritually speaking. And this is a piece of research from about a decade ago where more than half of the adults here in the United States apparently embrace the idea that there is a conflict between Scripture and science. And anywhere between a quarter and a third of young adults who are Christian embrace that same idea. They see there is a conflict there between real science and what the Scripture says. And even some of them consider that we as Christians and churches are out of step with where science is, and we should do an upgrade of sorts. When Darwin went on his famous voyage around the world, he kept a journal, and that was the basis of The Origin of Species that published decades later in 1859. And the earliest tree of life that you can find in his writings is this sketch on his journal, captioned with, "I think." And then he developed this tree here with some branches and some lettering, but it was a primitive germinal idea. It didn't take long, though, for that. You see how simple this tree is here. Look at this one. A few years later, Ernst Haeckel, this was a German scientist, and he has been nicknamed Darwin's bulldog because he was very militant, very passionate, very prolific. He traveled everywhere. He came with this complex, fascinating tree of life, as you hear, very structured, very complicated, very impressive. When he was late in his career and he published this book, Last Words on Evolution, Haeckel did, he summarized the understanding that they had of the human cell and the beginning of the human being as that initial cell as a simple globular protoplasm, like a blob of jello. That was what they envisioned the cell was, surrounded by a membrane, little, 1/120th of an inch in diameter, and there is some nucleus inside. That was the state of knowledge at that time. And here we are, 165 years later, as I said. And we know more, don't we? I mean, this thing that you see here is the molecular engine. It's an artist's representation, but tries to keep things to scale. These are all proteins and pieces in a machine that is functioning non-stop in our mitochondria. All the cells in our body have these energy-producing machines in them. We can have 5,000 of these in one mitochondria. And in a heart cell, you can have 5,000 of mitochondria per cell. So you're talking about, 25 million per cell of these machines that produce energy. These little sparkles that they show around here is what results from this machine. What goes in is the oxygen we breathe and the hydrogen that comes from the food that we eat, and what comes out is the ATP, the energy currency for every single cell in our body. So, like I said, we have millions of this in every cell, and they look complex. They don't look like a blob with jello, right? And then inside our cells, there's other things. This is an animation of a structure that transports a vacuole with substance that the cell produces, and it needs to go somewhere else. And the cell shuttles it out, building in real time a highway, literally, of tubules that this transporter walks on to carry it where it needs to go. And then the highway is broken down and used somewhere else. It doesn't stay there all the time. So it's fair to say that the structure of the cell that we understand today, and we haven't gotten to the end of it by any means, is like a city where we have city limits in that membrane, which is very complex. I remember I was in the second year of medical school in Romania, and there was a very militant evolutionist histologist that taught us a lesson, a lecture on the cell membrane. And he started to show us the bilipid layer and how those lipids, they are not sitting there dead. They move, they flip-flop all the time, they tremble, they organize. And then you have proteins traversing the membrane with receptors, with all kinds of complexities. And at the end of the lecture, we were all in awe about, wow, this thing is so complex, it's amazing. And then he ended by deflating our balloon, saying, we cannot accept a designer for something like this. As compelling as it may look that somebody put this together, it just came to be. We cannot go there. By our commitment to naturalism, we have to rule that out from the start. But we don't have to, do we? That's the whole point I'm trying to make today. And those vesicles that you saw traveling with precious material going from one place to the other, or even outside the cell to go somewhere else, like hormones, for example. You have a postal service, in this thing, where things get tagged, marked, and sent places. You have the ribosome. We'll talk about this in a minute. These little tiny things that we have, millions of them sometimes in a cell, these guys make protein. They are manufacturing plants for proteins. And of course the mitochondrion, which makes the energy, that machinery that you saw, and a bunch of other things, including the most important of all, perhaps the nucleus, where the DNA, the genetic material is located, right? We have come a long way from the blob of jello. So is Darwin's idea still standing? Obviously it is. And one of the reasons, the main reasons why it's still standing and doing well everywhere, is because of these three guys, Fisher, Haldane, and Wright, the evolution's trinity as they are known. You see, back in Darwin's day, nobody knew about DNA genetics. It was not discovered yet. He had some ideas of his own, and there was a monk, Mendel, you may have heard, who was discovering the laws of heritability. And apparently there was even a little bit of a correspondence maybe, or at least Darwin had Mendel's ideas on his desk that he could look over, but he kind of didn't buy into it. He came with his own ideas that didn't turn out to be true about heritability and how genetics work. But by 1930s, it was clear Mendel was right. And it was clear genetics is what determines how we look, what creature comes out of that initial cell. So they had to reconcile the idea of Darwin with how genetics work. So these three guys did it. And the way they did it, and they came up with this the new Darwinian synthesis, or the modern synthesis. They said, listen, what Darwin talked about when he said we need numerous, small, successive modifications, those are the random mutations that happen in DNA. And those mutations are being worked on by natural selection, and this creates the new life forms over a long time. And that's how they reconciled that and made it acceptable in the modern era of molecular biology. And this is how, to this day, you have, if you watch any documentary, have you noticed at the beginning, I jokingly call it Darwin worship, they have to present some sort of, millions of years ago, things came about and, natural selection worked on it and evolved it and so forth. But it's also present in schools, in academia, in the media, of course, and even churches. If you look at the Catholic Church, for example, who's one of the larger Christian denominations on this planet, the way they struggle to and strive to incorporate the ideas of evolution is quite amazing in their doctrine and understanding of, for example, was Adam and Eve, were they real people, or were they some hominins that God at some point tried to put his spirit in them? Imagine you want to be a cook for a day. You look for a recipe that you don't have at home, you don't have in your kitchen. You have to go to the library. You have to check the book out, but they don't give you the book because they are afraid you're going to mess it up in your kitchen. So they say you can take a copy of the recipe that you need to make and take it home, and then you can make your cookie or whatever you're trying to cook home from that copy. This is how genetics work in a way. The library, where the book is the nucleus with the genome. We have 3 billion letters that spell out the human genome. I mean, they're pairs, so in total there are 6 billion, but 3 billion unique letters. And then a gene is the recipe that you get. And in order to make the cookie from the recipe, you have to take it out, like I just said, copy it first from the library. And this is what this first step does. You transcribe the information from the recipe, from the DNA, from the nucleus, and you make this messenger RNA. This is the copy of the recipe that you take out. And then you bring into your kitchen, in the cytoplasm, outside the nucleus, and translation happens there. Basically, you read the recipe, you look at it as a cook, and you say, okay, I need this and this and this and this. This is what the ribosome does. Okay, it takes that copy of the protein template, it reads it, and then sends this guy, the transport RNA, is the guy that goes shopping. You need ingredients for the recipe. So he goes and gets this amino acid and the other, and brings them to the ribosome in the kitchen to make the ingredients fit together, and finally you have the assembly of the protein. And this is how genetics at the very basic level work. People realized that in humans, as well as in most other organisms, there is only about 20, maybe 30,000 protein coding genes. In other words, in the whole cookbook, you only have 20, 30,000 recipes that code specifically for a specific meal, okay? But this only represents 1.5%, maybe 2% of the whole text in the book. 98% or more is not coding for protein, it's just sitting there. And in that context, if you had an evolutionary mindset, this was a godsend. This was actually in the 60s that this concept was developed, even before the Human Genome Project was finished. So this is where the concept of the junk DNA was developed, in the sense that they said, listen, we have over 98% of the genetic letters not specifying anything in particular. And therefore, this is junk. This is the sandbox of evolution, because you can have mutations popping in there like in a cauldron boiling randomly all the time. And ever so often, in millions and billions of years, you can have something good popping out of there, and the organism adopts it and evolves over time into something better and entirely different over time. And that was the concept of the junk DNA, which is a very dear concept in the evolutionary community. I'm going to show you a timeline of that. This is in the 60s, late 60s. A Berkeley professor here in California, I believe, in the US, he was confident that 99% of DNA is junk. It's not doing anything. A few years later, by the 1980s, when they started to understand more about DNA and how genetics work, things became a little less convincing, but they were still pushing this idea. This is Francis Crick. This is one of the co-discoverers of the structure of the DNA. Famous guy. I think he got a Nobel Prize for this. Same idea. Much of the DNA in our organism is little better than junk. Here's Francis Collins. This guy was the director of Human Genome Project. He's a Christian, interestingly, but he's a theistic evolutionist Christian. He tries to to marry evolution with Christianity. So what he was saying in 2006, says he's toned it down. By that time, understanding and science progressed enough to understand that it's not that simple. So he said, well, 45% is probably nothing more than flotsam and jetsam, genetic junk. But you still have the very militant branch of evolutionists like Richard Dawkins. He's like the modern day Haeckel. He's still, years after, Francis Collins was saying this in 2006. 2009, Dawkins is still staying at 95%. He's not budging. He said, no, we are still 95% junk. And then 2013, I think it was, when the ENCODE project published its findings. You see, there was this international group of researchers that joined forces throughout the world, and they said, let's each of us look at one piece of the genetic code and see what it does, if anything. And they did their experiments. They looked to see if anything gets transcribed, anything gets checked out from the library for that segment of the book, for anybody interested in that text or not. And what they found was astounding, is that over 80%, and chances are it's more than that, they did what they could with the tools they had at the time, gets transcribed. It gets checked out from the library. It's functional, essentially, because the body, the cell, even with an evolutionary mindset where you don't do nonsense stuff because you lose energy, you become less fit. If you do something, it must have some purpose, right? 80%. So jumping from 1.5% to 80%, pretty big jump. A lot of people were very mad that these guys published all their research at once instead of one article at a time that could be dismantled and attacked with the strategies that they unfortunately use very efficiently. And there was a lot of panic in that community. And in fact, even one of the main researchers there made this statement saying that the big surprise was not only that almost all the DNA is used, but also that a large proportion of his genes were switches. And I'm going to go quickly through this in the interest of time. But this was the old genetic dogma, molecular biology dogma, where you have DNA copied into RNA, protein, and you get then the cookie at the end, right? The new one kind of goes around the same pattern, but you have this big thing here, which we didn't acknowledge before, which is the control element. Because think about it, if about the same 20, 30,000 gene coding proteins are similar in mice and humans and elephants and whatever animal, more complex animal you can think of, why are we so different? Why are humans capable of art and culture and music? Like we heard these beautiful songs here, Glee can play the violin like it touches your soul. And you look at a dog, I mean, it's great, it relates to you, but you can never teach a dog to do that, right? And yet they have the same protein coding genes that we do. What makes the difference? Well, the difference makes this other 98% of DNA, where in humans, it's like you have an instruction book with how to put these Lego pieces together in such a way, not just physically, but even behaviorally and mentally, that you get a human capable of spirituality and even grasping concepts like this today, as opposed to any other animal. So this was the big discovery in the ENCODE project, that there's a lot of control elements that are necessary, important there, and you cannot just discard it as junk. But I want to think of something. Had people in the 60s, when they realized that there's a lot of DNA not doing direct protein coding, had they not labeled it junk in 60, 70 years of research, don't you think we would have been much further along in understanding, in science, in medicine, in disease control, had we studied intensively that thing for 60-some years, instead of thinking, oh, it's junk, let's just let evolution do its thing there, there's nothing to see there. You see, it matters which perspective you come from. At least some of them they are honest. Francis Collins, remember he said 45% was flotsam and jetsam. In 2010, after more data came out and the ENCODE project was being worked on and he was an insider and he knew what was coming, he said, if you thought, this is another book he published, The Language of Life, if you thought that DNA molecule the DNA molecule comprised thousands of genes, but far more junk DNA, think again. These are trends that happen behind the scenes. You know, if you look in the literature, and one of the reasons I'm presenting you this information is to get a glimpse of what's happening under the surface, because what's presenting in the front, what's presented in the front, is a very unified, there's no cracks in this, we figured this out, it's all proven, don't even think about questioning it, okay? Where in fact, when you read what they say, and sometimes they get honest and say, like Francis Collins here, the discoveries of the past decade, little known to most of the public. There isn't an effort to make this go into the high school, you know, like he says here, have completely overturned much of what used to be taught in high school biology. Well, here's a screenshot of my son's biology course on evolution. And one of the proofs that they present, these are drawings that Haeckel, Darwin's bulldog, put together back 100 and some years ago to try to prove that very early in life, different animals, when they are at the very, very early stages of their development, the embryos look the same. And he made the case that even they had gill slits, suggesting or making the statement that these were gills, we were fish, all of us. And you probably remember this when you were in school, probably, right? I remember this when I was in med school in Romania. You know, we all had gills when we were embryos, and that's a sign that at some point our ancestors were all fish. These are no gills. These are folds in skin, okay? In fish, they become gills. In humans, they become ears and jaws and neck and muscles. But they are little folds. But more importantly, these guys are not reflecting reality. These were doctored images in a major way to make them look similar. And this has been acknowledged. You know, 2015, for example, in the New Scientist, they openly acknowledge, and books have been written, articles have been, you know, they state in this article that it's embarrassing but true. Some of the most influential drawings in the history of biology are wrong, exaggerated to fit a thesis, you know. And yet, here they are, still 2024, presented as proof that evolution is true. You would think that this would be taken out, and it drives me a little bit mad to see this, you know, in our children's education. Now, spiritual break here. Can you guys think of a place in the Bible, a text that conveys this idea that something that was considered worthless, useless, became central? Indeed. Psalm 118, and I think other places, Isaiah, New Testament, the stone which the builders rejected, they thought it was junk, turns out to be the most important thing. The same thing in junk DNA. The modern synthesis, the new theory of how evolution works in this DNA age with the advances in molecular biology and in genetics. Basically, you have these ingredients where you have natural selection, you have those mutations in junk DNA, supposedly, you need long time, and some other things. They talk about genetic flow, genetic drift, and so forth. But mutations are by far the most necessary, important ingredients. That's where the novelty comes from, right? That you need. And then you have new forms of life. And I think it's important to understand that at the foundation of this, as I hinted before, is this methodological naturalism or materialism. This is not a scientific finding that we did an experiment we came to understand. This is a philosophical commitment. It's a doctrine. Doctrines are religion elements, right? They are not scientific elements. You don't learn doctrine in science, generally. You can have postulates, but you don't have philosophical doctrines. And yet this is the foundation of evolutionary science, because it's important for that to be there to rule out divine intelligence as a causal factor. That's where they start from. And you'll see this. I'm putting a couple of quotes here. This is one famous one from Darwin. He removed the creator, and instead he says, there's this force, natural selection, out there. But it's like God, because what is it doing? Is he envisioned it as daily, even hourly, scrutinizing, okay? When you scrutinize something, you have intelligence, right? I mean, there has to be some algorithm there where you scrutinize things with. You see that? This is personifying and deifying natural selection. It's basically some form of godlike entity that's scrutinizing throughout the whole world, every variation, even the slightest. And what does it do? Another intelligence requiring thing, rejecting which is bad, preserving, adding up, right? Silently, insensibly working everywhere, the improvement organ. And here's another one. This is a more modern promoter of evolution, relatively speaking, to Darwin in the 40s, Julian Huxley. He essentially said it. He said, natural selection is a power more majestic and subtle than any mythic god. And you need something like that to create. I mean, that's obvious. And one of the reasons I want you to think about this is to understand how is it possibly logically for a scholar, for an intelligent person in today's age, to embrace evolution where design is so obvious. This is how you make this commitment that there is this entity, natural selection, that is like a god, and then everything becomes possible. And in fact, one of the more striking statements from their community from '97, from this gentleman, Richard Lewontin, an evolutionary biologist and writer. He wrote a review of Carl Sagan's book, The Demon-Haunted World, in '97. And it's important to look this up if you cannot read it here. I'm not going to read the whole thing in interest of time, but I'm going to underline a couple of words he says here. He starts by saying, we take the side of science. See, everything is presented as pure science, although the basis we saw is philosophical doctrine of materialism. In spite of all these counter arguments that we see, we even tolerate just so stories, okay? We are okay with that. We realize they are just so stories. We realize they look kind of stupid, counterintuitive. You know, look at these words here. No matter how mystifying to the uninitiated, it doesn't make sense. And yet we embrace all of this. And we embrace it in an absolute way, this commitment to materialism, because the whole point of this thing is we cannot allow a divine foot in the door. So this is where we start. We start by, we want God out of the picture, period. Absolutely, there's no place for that. And therefore, we can put up with all this nonsense, all these things that doesn't seem to make sense, because one day we'll figure it out, perhaps. So we'll be patient. So, just to make this more clear, my son helped me make this graph yesterday. So, in the scientific method, you've been in school, you have kids in school, what's the usual sequence? You come across an observation, triggers your curiosity as a scientist. You ask a question. You do some research. You form a hypothesis to explain that thing. You then do an experiment to see if it turns out the way your hypothesis goes or against it. Either way, it's fine. You at least know whether it's that or the other. And then you analyze the result and you draw a conclusion, right? Those are the typical steps of the scientific method. But in evolution science, it's a bit different. You start with this. As you've seen, I'm not saying it. I gave you those codes because that's where they start. When you get to the bottom of it, when you talk to these people, this is the foundation. And then they ask the question, they form the hypothesis. But all of these things happen millions, hundreds of thousands, billions of years ago. You can't quite put together experiments in the vast majority of cases. It's not possible for them to show experimentally what they say. And to replace this important, crucial element in the scientific method, they write a story, okay? They publish it as fact. Even in high school, as you saw there's a story with the embryo that Haeckel put together. Facts didn't matter. And then they draw the conclusions and present, this is it, science. But it's really not quite science. This is the core, and I'll make it short, of my response to Darwin's challenge. Remember what he said in the beginning. If some organ or structure could not have arisen through numerous successive and slight modifications, then my theory would absolutely fall apart. So I'll submit to you an example of that. The ribosome. This is the protein-making machine in each and one of our, or most cells that make protein, or all cells that make protein for that matter. And we have millions of them in every cell. They are tiny. They have two parts. They have this larger subunit and this smaller one here. And what you see here in orange, this little band, is the copy of the recipe, the messenger RNA that comes from the nucleus with the information how to build the protein. These two units, when they don't work, they are separate. But when the messenger RNA comes along, they combine together, they kind of pinch this template of messenger RNA, and they have a decoding structure in here that reads it. This advances step by step, 3 bases at a time, as they illustrate here. See that? And as this advances, the shopping version of the RNA, the transport, brings the corresponding amino acid that is coded by these little structures here, and they get connected here next to each other, and the protein is being built. You see how that gets built in here? That's how proteins are made, in a very simplistic way. Now, there is a reason I chose the ribosome for this example, because it really presents a chicken and egg dilemma for evolution. Because the ribosome units themselves are made of protein. There's 80 proteins making those things, plus a few RNA molecules, but mostly protein. And what makes proteins? Ribosomes. You cannot make proteins without the ribosome. And yet the ribosome is made of protein. You understand where the chicken and egg problem comes from, right? And more interestingly than that, all those proteins, each one of them almost is coded on a different chromosome. So you have to gather information. It's like going to, you have 23 pairs of chromosomes. You want to make the recipe for a ribosome, you have to go to 23 different libraries and get copies of instructions how to make it. It's not like it's all in one place, easy, let's get it out and make it, boom. So it's quite complex. God had fun with this, I think, when he created it, just to make it hard. And here's this article that I submit to you here from The Origin and the Evolution of the Ribosome. This gentleman, George Fox, is a famous name in evolutionary biology. He still works at the University of Houston, I think, in Texas. And he specializes in trying to figure out how ribosomes came to be. Because you can't have life without ribosomes. You know, there's no way. And yet, you need ribosome to make the ribosome. This is one of the landmark studies in their literature on the origin and the evolution of the ribosome. So I want you to pay attention with me. It's technical language. I'm not going to burden you with trying to understand everything he says. I want you to pay attention to the narrative that is being built at the beginning of that explanation. He's giving a timeline of how the ribosome works. This gives you an idea, again, an artist's representation, like an animation, but kind of true to scale, as it were, on how the messenger RNA is being read, how the transport RNA brings the amino acids and they get exited, and the protein is being built behind the scenes here in real time. And some proteins have thousands of amino acids. This is like a complex machine. It's not something you can cobble together easily. This is very representative of what the science of evolution in the specialty journals does, how the science works. So you don't think I made that thing up earlier when I said there's no God and there's a just so story instead of an experiment. Pay attention to that framework as you read this with me. So they envisioned this ribosome that it started to emerge in the RNA world. This is the most popular origin of life theory in the non-Christian environment now, where they think RNA was the first thing that came about. And obviously the ribosome should have arisen from that world. And then he builds the narrative starting saying, when these RNA, there were little RNA molecules around floating there, they encountered each other. You know, in the presence of an RNA ancestral to the peptidyl transfer center. This is this area where that messenger RNA is being read. That's the PTC right here. There's a tunnel between the two units through which that messenger RNA goes and is being read, the information off of it. So he needs to figure out how that came to be. So he envisions these little RNA molecules. They found each other somehow in that original RNA world. They encountered each other. And there was some ancestral other piece of RNA that kind of looked like this somehow. And then they started to form little protein, 3 protein bonds, amide bonds there. That resulted in larger peptide-like molecules being created. And here's the only evidence in the whole article where they show that such bonds between amino acids could potentially be formed in an RNA world. This is the only evidence in the whole article. And the evidence is in the realm of possibility. It's not really clear-cut that that can happen in this specific scenario, but it's in the realm of possibility. And then they need some magnesium. Now, here's another problem. The peptide, the early protein being formed there, would be a mixed chirality, but enriched for the L version of amino acids. What does that mean? In all life, molecules, proteins, have a three-dimensional structure. And they have a left version and a live version. There's an L and a D version. Just like the hands or any 3D object in the mirror, you can create the mirror image. And in nature, they get produced similarly, 50-50 usually, when you have a reaction that doesn't care about that aspect. But the problem is, if you do that in life, and you have a receptor, for example, for a protein that has to fit like the key in the lock, if you make a mirror image of your key from your house, would it work? You won't. So you have to have only that form, so you would waste 50% of your work if you made both copies, left and right, all the time. So God, in his wisdom, decided to make only the left version of all molecules that we are made of and any life form is made of. We have only the L version. So he needs to explain that. So he's thinking that perhaps the L version was enriched. Perhaps. Okay, here's a great experimental evidence. Perhaps. As a result of an excess of D-ribose. Let's focus on the words. Something might stabilize. Is that a result of an experiment, I ask you? Now, here's a good one. As complexity increased, you know, we had some dead branches in our yard that the snow caught us without being able to get rid of them. So they are all covered in snow. Random pieces, big, small, cut, uncut. Is it reasonable for us to expect that by springtime, when the snow melts, say, we're going to see a little structure nicely built from those branches as complexity increased over winter? Is that how things work in nature? No. So they throw these things in there, and it drives me a little mad when I read this thing, because yes, it's a narrative, and they acknowledge it's a just so story. But then when you are a kid and you go through school and you mature and you read what they present in those, this is not shown. This is behind the scenes in the specialty literature. In the biology textbook, it's a myth that the theory of evolution is just a theory. It's beyond that stage. Everybody accepted it's true. The evidence is overwhelming. And yet this is the type of evidence as complexity increased. Who increased it? Where is there an experiment that shows in this situation that complexity increases? No, there isn't. There's nothing like that. You know, and then here's another one. The single domain transport RNAs and the PTC region, this is the crucial elements of this whole process, would emerge. So wait a minute, how would that emerge? You know, it's like, you know, abracadabra, they would emerge. Literally, that's what this is. So it's absolutely incredible. Here's a statement that's important to acknowledge, although there is no evidence currently addressing this, okay? So when it comes to experimental evidence, to data that you can reproduce and show, this is how it happened, I can show you, I put this 2, 3, 500 things together, and it leads to what I'm saying here. No, and they are fine with saying there's absolutely no evidence addressing this, but then they go on to explain how the story went, how these things put themselves together. Have you ever seen one of those stop motion animations? We were looking at it yesterday with, I was tempted to put one, but I didn't because of copyright stuff, where people put together car engines. You know, they start with a crankshaft and whatever, and they put different things, but they make it a stop animation, so it seems like it assembles itself, right? That's kind of the narrative that is being built here. It's basically stuff that just, you know, moving this there, the growing small unit. Well, how is it growing? You know, It's like you are watching this develop into this marvelous thing, and you think it just happened by itself. Essentially, that's what this whole narrative is. The article is much longer, but it goes along the same lines. I will stop here because it's late. But it's like this, just to illustrate it another way. When the first horse-drawn carriages, they were quite fancy toward the end of their golden age. But it didn't stop there. You know, one day we had one that kind of looks similar, but it has an engine inside. You don't need a horse anymore, okay? It evolves. There was a side branch under the constraints of maybe Arctic conditions where the wheels were replaced by that sled, but it didn't evolve much further from there as far as I know. But this thing continued to evolve. It got fancier, it got lights, it got comfortable seats, it got the cover for rain, it got the rubber wheels, and then you got really aerodynamic shapes and all kinds of gizmos, and pretty much is ready to take flight. So let me ask you, as you look at this is indeed the evolution of the automobile, right? You agree with me? We started here and we ended up there, and who knows, in another 1,000 years where we might be, right? But the key question here, how did this evolve? Did they just morph into each other as they put on more mileage and more mileage every year, and more cars went on the road, and one day you went to work with this and you came back home with that? Is that how it worked? It sounds ridiculous, isn't it? But you have to have what for this to happen? You have a designer, engineers, experts in, mechanics and fluids and materials and aerodynamics and a million things, with a computerized version of it, IT people and all of that, right? Otherwise, none of this happens. In fact, the more you drive it, the more what happens eventually? It falls apart, it gets broken, you need to fix it, you get another one, right? And yet, sadly enough, the majority of the scientific journals that attempt to explain how evolution works, not in just ribosomes like I explained you earlier, but in most other things, follow this pattern where they present comparatively stuff. Look, this is in what they think it's early and later, and it must have just, evolved from one to the other, removing God from the picture in a very deliberate way. Anyways, we started with Romans 1:20, and I just want to draw your attention back to that text. This is very important, where Paul says, since the creation of the world, God's invisible attributes are clearly seen. We have to remember that and be bold about it, I think, as Christians. These are clearly seen inferences of design. It's obvious. Even more so in the modern world, with those things that we see now in the cell, being understood by the things that are made. Now, the word that Paul uses here, made, I took it from the Strong's Concordance lexicon here. It's interesting. That specific word has to do with what? Workmanship. This is not just some stuff that got coupled together. There's an elegance, there's an artistry, there's, watch this, craftsmanship or artistry involved in the creation of this thing. And there's beauty there. We all are aware of that. We can see it, we can sense it. And as a church, we have the three angels here. I think they are better than the ones I put together here that you have on your church. And the first angel's message, what does it say? Flying mist of heaven, the everlasting gospel to preach to everybody basically on earth. And what is he saying with a loud voice? He says, fear God and give him glory, for the hour of his judgment has come, and worship him who, the same word used here in Greek, like I just showed you, with the workmanship, the artisan elements and everything there. Worship him who made the heaven and the earth, the sea, and the springs of water. And as a Seventh-day Adventist church, historically and to this day, we trace our raison d'etre, or the reason we're here, the mission that we are to accomplish in this world, among the many thousands Christian denominations. Why do we need the Seventh-day Adventist church? As we understand it, our certificate of birth, so to speak, has to do very much with the messages of these three angels, one of which is this: We have to be able to present to the world a convincing case that there is a creator who created in a very beautiful way, in a very obvious way, and we're not here because we are the results of some chance events over billions of years of age. And that was the message I want to bring to you today, and I hope it resonated with some of you.